In our study, we compared ctDNA and tissue-based genomic testing in metastatic renal cell carcinoma, and we aim to explore the temporal impact between blood and tissue by using a 112 patient cohort. We used a commercially available ctDNA platform that assesses 73 to 74 cancer genes. And for tissue-based testing, we used either whole exome sequencing platform or targeted next-generation sequencing. We only included alterations that were covered by both assays, and our results show that 32% of alterations that were detected in tissue were also detected in ctDNA. And this proportion increased to 43% when we looked at samples that were collected within 6 months. And it increased further to 51% when the time elapsed between these assays was just less than 3 months. But when we looked at samples that were collected more than 6 months, this proportion was just only 21%. And in conclusion, our study shows that ctDNA and tissue-based genomic testing continue to provide consistently high levels of agreement, and we show that tumors evolve over time, and ctDNA is a great tool to capture this. Most commonly asked question is the possible contributors of discordance when we looked at ctDNA and tissue-based genomic profiling. And the major causes of discordance is the temporal and spatial heterogeneity. As it is well known that tumors evolve over time, and also we discussed in our study, temporal heterogeneity is a major contributor. And for spatial heterogeneity, tissue-based genomic profiling is perfect at detecting the alterations that is located in the sample.